Hello, everyone. This is Arson. I'm IMARIS sales and application engineer based in Taiwan. Today, I want to introduce the IMARIS software, which is the ideal solution to interactively analyze microscopy images for you. So, what is IMARIS? IMARIS is a software for specialized for 3D and 4D image visualization and the quantification of microscopy images. Also, IMARIS provides the tools for you to share your research results, and also IMARIS with a team of people that are here to help you all along your image analysis path. So I'm Arsen again, and this is my email. If you have any questions about IMARIS, you are welcome to contact me through email. Okay, so first, we start with the visualization and analysis part. So usually to present IMARIS main functions, I will follow a image analysis pipeline with a few applications, starting from how to import your raw data to IMARIS and do some pre-processing before the visualization. And after visualization, if you need to extract some numbers for the structures or objects you are interested, then we can go through the detect and analyze step. And in the end, if the default analysis functions cannot satisfy your research purpose, we also have a customization functions which allows IMARIS to talk to other software like MATLAB or Python to customize your own analysis protocol. So what kind of images can we import in IMARIS, and what kind of image format? IMARIS was first developed in about 30 years ago and started from the fluorescence images. And since then, IMARIS was largely developed so that nowadays it can also open non-fluorescence images like historically standard slides and electron microscopy images, etc. But of course, the majority of IMARIS users they are still based on the fluorescence microscope. And in terms of image formats, IMARIS has more than 50 readers so that it can convert automatically and open all commonly used image formats, including microscopy systems from Zeiss, Leica, Nikon, Olympus, and other manufacturers. And if the raw data format is not supported by IMARIS, at least you can convert it to TIFF first. Then from the TIFF format, IMARIS can convert to .ims IMARIS format successfully without problems. And in this slide, I'm showing the reconstruction of the developing zebrafish embryo image of 1 terabyte. So there is basically no limitation of the file size you can open and visualize in IMARIS with your computer. So once your image is imported to IMARIS as a .ims file format, depending on the situation, you might need to prepare it for the optimal visualization. So here in IMARIS, we have some built-in pre-processing tools for fluorescence images like smoothing, background subtraction, also deconvolution. And if you are working with a large data set, we have a standalone program called IMARIS Stitcher, which can help you stitch the tile image into the whole sample so you can then bring it to IMARIS for visualization and analysis. Also in IMARIS, we have this align function, allows you to present multiple images in a single single working space. So you can do applications like correlative analysis or overlap two individual images for comparison. For deconvolution, it is actually a software algorithm to improve the image quality for visualization and analysis. We can use format images from microscopy systems like wide field, confocal, spinning disk, turf, etc. for deconvolution. And it contains two different algorithms. One is faster, another is slower, but is quantitative. And as you can see from this image, before and after the deconvolution, you can see it actually improves the resolution and the signal to noise ratio. Our deconvolution algorithm is called Clearview, and it's GPU accelerated, so it's quite fast, and it's batchable, which means you can run the deconvolution algorithm on multiple images automatically in IMARIS. For stitch functions, we use a software called IMARIS Stitcher, and IMARIS Stitcher is quite powerful. It has a capability, it is capable of doing the stitching for very large data set over several terabytes. For example, like these brain images, this is 56 tiles of 73 gigabyte each, and total size is about 3.5 terabytes. IMARIS Stitcher can do the alignment quite accurate in the X, Y, and the Z direction, and very easy to use. For the alignment function, as you can see here are a few examples. First example is the overlap between electron microscopy images over fluorescence microscopy images. This is called a correlative microscopy. You can see the overlap and verify the different maybe subcellular components with fluorescence microscope images and electron microscopy images. Another example would be high resolution Z-stack overlap with the low resolution XY image, 2D images with the whole sample field of view. So not only you can see the whole shape or whole scenario of the sample, but when you zoom in into a specific region, you can see the high resolution details with the analysis result. This example is two individually taken part of the brain images. We can manually align them together and present as a whole brain in IMARIS. The last example is the comparison between two type of sample images, mutant brain and the wild type brain. And you can see or compare the difference in the optical system very easily, like this. So after we prepare our data and ready for visualization, there are multiple ways to render the intensity voxels in 3D in IMARIS. So IMARIS have three different rendering mode, including the MIP mode on the left shows the transparent effect of the whole sample. So this is a small mouse embryo. Under the MIP mode, you can see clear like the blood vessel structure and the neuron, etc. inside. And on the right-hand side, it's called the blend opaque mode. It take into consideration the transparency. So if you want to show a more solid 3D structure or 3D shape, then this might be a good choice because now you see the shape of this mouse, small mouse embryo. And furthermore, if you want to present the surface details of this mouse embryo, in the middle, we have this normal shading mode, which introduces a pseudo light source So when the light source shining on the surface of the 3D object, you can see the details of the surface. And we can use this mode in either a snapshot or create animation. IMARIS also have more visualization tools to explore for you to explore your data set. For instance, the also slicer allows you to show a 2D projection of your image in the 3D space. And there is a tool called the clipping plane. Clipping plane is allows you to cut the structure in any spatial orientation to visualize the inner parts. And we can combine all these tools to create a nice informative movies. And finally, we have also, I also want to mention, we also have this annotation tool, for a snapshot or make an animation to explain the details of this structure. Okay, so once you better understand your structure of interest, probably the next step would be to perform some quantification analysis. To do quantification analysis, first we need to detect the structure of interest from the data set you are studying so that you can extract the statistic in order to rationalize the particular biological effects you are studying. And IMARIS also provides a plotting tool for you to interpret your analysis results. In IMARIS, there are 4 segmentation tools, including spots, surface, cell, and the filament. To choose the appropriate ones, you must look at your structure of interest in terms of shape. First, the spot. Spots are useful to perform a quick count of roundish spherical spot-like shapes. For example, in this example, the nuclear staining in blue, if we want to count the number of these cells, we can very easily use spot tools to detect the intensity center of these nucleus and count the numbers. Once the spot is created, we can extract the numbers like statistic, like number counting, position information, diameter, intensity, distance, etc. Over more than 30 different statistics we can extract. And all these results we can export to Excel worksheet format or a text file automatically for further analysis. Yeah. This is the same for all IMARIS tools I will present later. So if the structure of interest is irregular shape or homogeneous stem like a cells, a nucleus, or the whole organ, then spots is not suitable to use for morphological analysis. In this case, we have to choose another tool called surface. Surface tool creates these mesh objects that you can then compare between themselves in terms of volume, intensity, and the morphology. As usual, all states are automatically calculated and can be exported into a pre-formatted file. Also, in IMARIS Vantage plotting interface, we can extract created objects from the image and, for instance, sort these nurse cell nuclei in a gallery according to the parameters of the spherical. Yeah. And in this HREM image of heart, this is an example where the automatic surface generation cannot satisfy the segmentation. So we need to draw the contours of all structures of interest in order to study the heart anatomy. So if the image is non-fluorescence, and it's difficult for surface automatic detection. There is also a manual way for our users to draw the contour manually in 3D space. And based on this contour we draw, IMARIS can still generate or create a surface for morphological analysis. Okay, so surface tool will extract not only intensities, counts, and positions, but also extract volumes and the morphological statistic. Then if you like to quantify the intracellular components relationships, meaning you would like to get not a number of spots for the whole image, but a number of vesicles per cells or a volume ratio between the cells and the nucleus, in this situation, you would pick the cell tool. So in this image, we have the blue cytoplasm staining can be detected as a surface, and the red nuclei inside the cells could be also detected as surface objects, and the green vesicles inside or outside nuclei could be detected as spots. Then IMARIS cell tool enables this global multi-object detection in one analysis, and it outputs statistics based on each cell. For example, the number of vesicles per cell or per nucleus, nuclei volume per cell, and here vesicles are color-coded according to their distance to the cell membrane. And in this example, we use IMARIS cells to segment these cells, even though we were not interested into the intracellular components relationship. But because IMARIS cell including this membrane detection algorithm, so even you don't have the cytoplasm staining, but you have a membrane staining, and you want to segment these cells, IMARIS cells is a good tool that you can use. So with the cell module, we can extract all previously mentioned statistics plus intracellular component relationships, and also has a membrane staining algorithm. And finally, a few examples of filamentous shapes detected using IMARIS filament tracer. Filament tracer was firstly developed for neuroscientists to detect neurons. It can extract multiple statistics to describe neurons, such as the number of neurons, which is the number of filaments, and the number of dendrites, their lengths, and the diameters. We can even detect spines and classify the spine according to their morphologies. And in this example, we can get the retinal neural network description using the dendrite length maximum for the axon length labeled in orange, and the dendrite length sum for the whole network length. And the dendrite straightness, number of branch points, number of terminal points would give a good idea of the network complexity. We can also perform show analysis. Also using IMARIS plugin, the convex hull IMARIS extension, we can extract the volume of the cell occupied in the 3D space. Also, here are more examples of filament tracer applications that don't fall on the neuroscience field. For example, if you have a vasculature images without lumen, you can get a full vasculature description. And in this example, bronchus could be detected and the filament tracer could extract their diameters. Chromosomes could be modeled as filament as well, and the green recombination size position could be measured using the dendrite length. So in the last example, some cilia were detected for density and the straightness analysis. So it's not limited to neural research, but also into the cell biology and other application field, as long as the shape is a filamentous structure. Okay, so filament tracer is useful to describe filamentous networks. So as you can see, the choice of the right detection tool to use is guided by both the shape of structure and the statistic you wish to extract. Also from version 9.6, the AI solutions for data analysis is now built in IMARIS. It has three different parts: AI segmentation, AI object classification, and the AI filament tracer. For AI segmentation, it is mainly for surface creation, surface tool. So as you can see, for some images which you find difficult for automatic surface detection, now you can just draw on the image the part you want to keep, the part you want to see as a background. In this example, green color is the one we want to segment, and the purple part is the background. And based on the examples you draw, IMARIS will try to segment the whole image for you automatically. And for object classifier in IMARIS, this is after the segmentation. So for example, on this image, we have all cells detected by spots. And if you want to classify these spots into different regions based on their background staining, then we can easily apply this machine learning classifier for this kind of classification purpose. Then after we classify them into different classes, we can further analysis, for example, the cell density or cell intensity difference between all these classes. And the AI filament tracer is included in IMARIS 10, which brings more potential for filament tracer for different kind of samples and the larger size of data. So it gives you a more accurate result, tracing result, and more type of different kind of samples. So if you are doing filamental structure related research, I suggest you to try this latest filament tracer. You may find it very helpful. Okay. So for those who is working with the time-lapse images, which means not only XY or XYZ, but including the time point, multiple time points, we can follow detected objects over time by these tracking functions in IMARIS. So with our tracking module, we can follow the morphological changes over time, or intensity change over time, position, speed, acceleration over time, etc. We can also get the trajectory properties like duration or length or straightness and more. In this example, this is a time-lapse zebrafish heart images with the blood cell detected using spots. So after these blood cells are detected over the time point, we can analyze its movement in IMARIS. And depending on the movement of the structure of interest, we can choose one of our 6 algorithms to perform the tracking. Another example of the tracking is the cell division. So this embryo cell division show you we can label spots and the tracks the way we need, for instance, according to their ancestry. And we can follow cell division, extract lineage trees, get the time of division, follow cell generation, and so on. So if we are interested in measuring the influence of cell division on a particular statistic, for example, the nucleus intensity change over cell division, we can plot a classical graph where along the x-axis we have the time in seconds, and along the y-axis is intensity. Then we can identify the intensity increase at each cell division time points. And here, what the special things IMARIS can do is it can synchronize the time points. So it synchronizes all the division events into these event tracks as a 0. So you can obviously observe the intensity change before and after the cell divided. So similarly to the previous examples where the spot objects were used to follow cell divisions, we can, in this example, track service object as well. Here, a killer T cells is eating the cancer cells, and the both were modeled as surface. And spot were used to track the cytotoxic granules, like in the red color. So if you are interested in measuring the granular behavior before and after the cell contact, both cell contact, we can set this specific fusion moment as a reference events here in the frame 17. Then we can easily plot, for example, speed of these granular spots change before and after the T cell contacting the cancer cell. In this example, we have cyan staining cancer cells, and the white color is the neutrophils. If we want to analyze, for example, the neutrophil intensity changes before it attacks the cancer cell, before and after it attacks the cancer cell, we can easily make this kind of plot to explain our discoveries in the IMARIS. Another example for tracking is the cytotoxic T lymphocytes movement against the tumoroid or experiment group. So in this time-lapse movie, we can track the movement of these T lymphocytes for X direction and compare the displacement in X direction between these two groups. We can easily find or easily observe the difference. So green color is the ones with the tumoroid. You can observe the T lymphocyte moving directions toward the negative X direction, which means it has a trend to move toward the tumoroid on the left-hand side, like here. And on the control group is just like equally move to the left and to the right. So there is no trend. Okay, so once the detection step is completed and the statistics are calculated, IMARIS can help you to go deeper in the analysis. For example, we have the tools to look at the colocalization of structures or classify the structure detected as objects, and even study interactions between structures, like affinities. So first, we can look at the colocalization problem for an object point of view, meaning we can create surface on each structure of interest and look at the volume of overlapping region between these two structures. The overlap volume and the ratio statistic are automatically calculated in IMARIS when these two surfaces are created. Sorry, let's go back. Okay. And we could be more interested by context between two structures rather than an overlapping volume. And this IMARS plugin or extension named Surface Contact Area can be used for this kind of purpose. And finally, we can look at the colocalization problem from the voxel perspective. To do so, IMARIS has colocalization dedicated interface where classically we can compute Pearson and Mendel's coefficients often required by reviewers and the percentage of colocalized voxels. For the classification, this is an example to study the cell migration in a particular region of interest of a brain. So we first have to define this region. And to get cell position relative to this region, we add a new reference frame, a new image origin point to the new position that has an anatomic meaning. And now we can classify all the cells into X classes along the X new direction. And for each class, we can not only get a cell count, but also get a sense of their, for example, density per class using the statistic named distance to nearest neighbors. So we can easily compare class densities within this image and later across multiple images. And this example is a 3D genomic analysis by DNA fish. In blue, we can see the nuclei and there are three different alleles in red, green, and the magenta color. So to get the count of all red alleles that colocalize with both green and magenta alleles, we will use an automatically calculated statistic named shortest distance spot to spot. So in here, we use two different parameters to classify these alleles. So we can easily find the red alleles which is colocalized with both green and magenta ones. So finally, in this planned cell image, there are three channels describe a multi-regional landscape. However, each region can really be described only using channel colocalizations. Probably each region can be described by its own texture, local and environmental channel intensities, and other unknown descriptions. However, here, at a glance, we could manually define these regions without really understanding what is going on. So in this situation, the new IMARIS machine learning classifier will enable us to quickly and easily classify all cells per region. To do so, we first detect all cells as spots. And to train the machine learning classifier, we select by eye 5 different sets of spots representative of 5 different regions. Then based on these selections, machine learning engine predicts for each other cells in which region it should fall. So 5 different regions are represented here by different colors. After a few optimization trainings, which meaning collection of wrongly classified cells, we get all cells correctly classified. So during this process, a new statistic are computed and ranked according to their weight. So this kind of statistic related to the machine learning classification protocol. Yeah. Okay. So just a moment. Okay, here. Let's go. Okay. Okay. Then to study the affinities between the multiple objects, we can firstly, for example, in this example, this is the organoid with different sample moving in and the cell moving in and moving out. We can firstly detect the organoid as surface and look at the accumulation of the blue signal over time inside the organoid. So this automatically generates a graph. So in here, just let me go back a little bit. So in here, x is the distance to the red organoid, where the 0 represents the age of the organoid, and the negative x values will be associated to blue intensities that accumulate inside the organoid, and the positive x values to blue intensities that are outside the organoid. So we can see very obviously inside intensity is much higher than the outside for the blue signal, which represent the cells. Then we can also model the blue cells as spots and study the attraction band of these cells to the organoid. So this time we will look at the probability density graph of spots inside and outside the organoid. After we detect the cell, blue cell as a spot, we can draw this plot for number of spots density. And further, we can compare this measurement with 100 stimulations of random distributions of spot represented by the dashed line. So we can actually simulate the random distribution result and compare it with the real data. And we can find the difference, obvious difference. For example, when the distance is below these 20 microns, we have much more significant number of the cells which is inside the organoid within this distance. So this is to study the affinities between these, for example, organoids and the cells. And you can apply it to any different kind of the objects you are interested in this kind of analysis using these plotting tools in IMARIS. So if the default analysis detection and analysis functions in IMARIS is not satisfied with your application needs, in IMARIS, we have these extension modules for customization. So you can expand IMARIS capabilities by integrating IMARIS extension plugins to, for example, write the scripts in MATLAB or Python, whatever programming language mixing your own and the IMARIS functions. So once integrated, extension appears as a button in the IMARIS interface like this. So you open the image in IMARIS, and you can click this button to send the image for example, MATLAB or Python for processing, then send back the result to IMARIS for further visualization and analysis. The previous example I showed in the filament tracer, the convex hull extension is working like this. So all existing extensions can be freely downloaded and used from the IMARIS Open webpage from our official website. So sometimes our user upload the script they created and share with other researchers with the similar applications. Then you can download and modify if you need. And as I mentioned, IMARIS can connect to Fiji and ImageJ and use the plugin directly. So if you want to use some useful plugins or for processing or analyze in ImageJ or Fiji, then you can just open the image in IMARIS and click the button. So IMARIS will send the image to Fiji or ImageJ for processing, then send back the result in IMARIS so you can do the visualization and further analysis. Okay, so that's about everything I want to introduce about IMARIS in case of the visualization and analysis functions. I also mentioned IMARIS also allows you to share your research work with your collaborators or other researchers. So what do I mean by sharing? Actually, it can be just sharing your results by presenting a movie created by IMARIS or a nice snapshot, which I already showed you a lot in the previous presentations. But also in IMARIS, if you want to share the data, I mean, not just the movie or snapshot, but the raw data, which you already, for example, processed in IMARIS as an IMARIS file format, then for your collaborators, they can download, for example, IMARIS Viewer. This is a free software and anyone can download from our official website and install on either Windows or Mac computer. With IMARIS Viewer, we can open IMARIS files with the segmented results and utilize all these slicing tools for visualization and make a snapshot very easily. The difference between IMARIS and the IMARIS Viewer is that IMARIS Viewer cannot output animation, and the IMARIS Viewer doesn't include any analysis functions. Also, we have this IMARIS file converter dedicated for converting the raw data into IMARIS file format. And finally, sharing can also mean sharing the IMARIS licenses. We have two different ways of IMARIS licensing. One is the node lock license, which the license is tied to a specific PC, so it can only be used on a single specific computer. Another licensing mechanism is called the floating network license, which we can use a computer as a host, and other remote PCs can acquire the license from the host via internet and use IMARIS on their own remote computers. Also, we provide multiple ways of supporting our customers, and also we regularly publish application notes on our websites. And if you are interested in learning IMARIS by yourself, on our website in the Learning Center, we have a lot of free resources, including the webinar recordings and a training series called IMARIS Homeschools, which gives a very detailed, thorough training of how to use IMARIS. And you also can find a lot of YouTube videos recorded by our customers from the training sessions, including the Chinese part from National Taiwan University and the English part from our customers in probably Australia and the other regions. Yeah, so we have a lot of resources you can utilize online to learn IMARIS by yourselves as well. Okay. So that's pretty much all the IMARIS instructions I want to present today. Yeah, and you can find some latest news from our website as well. So thank you.
Thank you.